to start the recording. So I do want to let you know that we will be recording this webinar, and later on the recording will be shared with all of you and those that were interested to participate and register for the program. To the hands-on biomedical data science. This online program is a course that combines user-friendly processing and machine learning methods designed for high-throughput, big biomedical data. And what we'll cover in this program is a combination of practical skills, how you can develop practical skills that you can later on use to address your own research and biomedical challenges that you're working on. We'll be talking about the various questions typical for the biomedical research industry, and we'll also be talking about how we can address the problem of big data, where computational resources and easy-to-use tools need to be combined in a seamless way to help us address biomedical challenges quicker. In the program, we'll use a variety of resources, and we'll talk today about how to get started, how to get set up, and also how to look at a variety of data sets that we'll be using throughout the program. Again, for those that are joining right now, welcome, and we look forward to getting to know you, and we're also expecting that we will have regular sessions where we can meet and address any questions that people might have and help everybody get started with hands-on biomedical data science so that you can be ready to solve biomedical challenges and analyze large data sets. And we'll talk about a few different aspects of that today. So in this program, in this short introduction, we will go through some of the logistics of the program itself, how to get started, how to get ready for the program itself, and start installing some of the useful tools that we'll be utilizing throughout this program. So the goal of this program is to understand how processing big data, analysis using machine learning, visualization, and different aspects of data analysis all fit together and are critical for our ability to understand biomedical challenges in the context of these high throughput data sets. Now, typically, we will look at the biomedical data challenges from the perspective of precision medicine, and we'll talk a little bit today about some of those challenges and some of those topics. The program is also designed to show a variety of different tools. We'll use at the Bioinfo platform, which is a multi-omics integration environment where you can process, explore, mine, and analyze data. And we'll look at also some of the other tools that will help you improve visualization techniques, understand how data can be analyzed, and we will also look at how we can easily review the data in tools like Excel, which is also a powerful spreadsheet application. So welcome everybody again. And in this introductory session, we'll review some of these topics and make sure that everybody can get started. So let's first of all talk about the schedule. The OmicsLogic Biomedical Data Science program is going to be divided into 5 different parts. So the first 4 parts are listed right here. We will start by introduction to the program, where we'll look at what high-throughput data experiments are, what are some of the challenges and opportunities associated with high-throughput data, such as next-generation sequencing, but also other types of high-throughput data. And we'll look at omics technologies, genomics, transcriptomics, metagenomics, epigenomics, so a variety of different omics that also include non-next-generation sequencing data, for example, screening of molecules, testing different drugs on cell lines, imaging technologies, and other. We'll also look at some of the challenges and opportunities in precision medicine, as well as associated with molecular data and drug discovery. Then we'll go into exploratory data analysis. So when we talk about exploratory data analysis, we'll be discussing how we can use statistical properties of data that help us summarize and compare a variety of different data sets. We'll talk about the challenges in preparing data, structuring the data so that it's accessible, but also making the data comparable so that we are not looking at the noise, but we're really comparing signal that is biologically relevant. We'll also look at a variety of tools that we have, like probability, correlation, and association. What do they mean? How do we quantify them? And how can we use that in different stages of our data analysis? We'll then look at the common data visualization techniques in Excel and R, because we would like to use those to work with our data sets and be able to interpret what we are finding. The third portion of the program will be dedicated to data mining. Mining large data sets usually involves utilizing unsupervised machine learning techniques, where we are going to be looking at prominent trends and variation that we can compare and identify both in big data sets, but also the features of the data sets that we'll be looking at. We'll also look at how we can use very specific methods like principal component analysis, dimensionality reduction, and how do we prepare the data using filtering, normalization, log transformation, and other methods. Then we'll move on to classification. In the context of discriminant analysis, when we want to compare a variety of data sets and identify how they are different, we can use a variety of machine learning tools. Typically, these are going to be supervised machine learning tools where we have an annotated data set. Using the annotation, we will be able to train a model that can later on be used for prediction. In other words, it can help us identify how the data set that we have trained the machine learning method on can help us further on identify the trends in a new data set. So we'll cover a variety of different methods: discriminant analysis, decision trees, random forests, support vector machines, and even talk about deep learning. But after this basic training, any one of you is going to be ready to apply these techniques to a project. And that is a critical part of the program. So we encourage all of you to, first of all, understand some of these methods, understand how they work together, and then start thinking about how can we apply these to new questions that you might have and to some of the exemplar projects that we already have on our platform. First of all, the schedule itself is listed already on our site. So there you can see all of the different sessions. So I went over kind of the bigger 5 distinct portions of the program. But if you're interested to see each individual session, you can go onto the OmicsLogic page and you'll be able to see all of the individual sessions. And also, we have already a number of exemplar projects that you can choose from, and all of those projects are going to be available to all of the participants of the program. So I'll go a little bit into how to register and how to make sure that you have access, but here's the code that you can use to get access to any of the projects that we have. Total fall, right here. So let's talk about the registration and how to get started with the program. So using this link, and I'm just going to paste this in chat, so please make sure that you can see that. Using this link, you can get to this page. On this page, you have to be logged in, so you will see your profile image right here. And then right here, you see today's session, program overview, as well as all of the following sessions by date. And you can register for that specific session. So look through some of these sessions, make sure that you registered for the upcoming ones. so that you can join those and you can get notifications about the materials for those sessions. Down here below, we have a forum, or essentially a stream of updates from the participants of this program. So you can look at everything that everybody is doing, and the goal of having an updated profile is so that this becomes a network that you can leverage throughout the course of the program and after that as well. So you can see that each person has a profile. You can click on that profile and you will be able to see the background of that person, potentially social media links, like link to their LinkedIn profile or something like that, and also see their activity. So the reason we have prepared this organization page is so that all of you can get to know each other, and we can easily share updates, for example, like this one, where we post important links and encourage people to ask questions or even answer those questions right here. Also, you have a number of links right here above. These links are going to help you navigate the various parts of the program. For example, you can see the curriculum page. So let's just go right here real quick. The curriculum page lists out all of the events by date. So you'll be able to see that today we are doing program overview, and gives you kind of a breakdown of some of the topics that we will be covering. Then you can see that we will go on and talk about next generation sequencing and various omics technologies. We will talk about summary statistics and what are those statistical properties, data visualization for exploratory analysis, factor regression analysis, how we can leverage factor regression and correlation between factors that affect what we see in the data, visualization and network analysis, dimensionality reduction, clustering, decision trees, etc. Essentially, up until we reach this stage when we start talking about projects. So all of these online resources are going to help with the program itself as it continues. But you can also start looking at some of the courses associated with the program itself. Especially, you can look at this tab right here called Courses, where we have already added a couple of courses that you can get started with. So I'm just going to go to this tab, and here you can see that once you enroll in a course, you will see a progress bar, and this progress bar shows you how far along are you in this course. And also you will be able to see some of the courses are paid, but again, Total Fall is that code that you can gain access to any of the online resources for free throughout the program itself. So let's review so far what we've covered. One, you have to be registered on the EDU website. You have to also register for the program. So once you get to this link, if you're not a member of this program, there's going to be a yellow button that says register for membership. And once you register for the membership, you're a part of the OmicsLogic Data Science program. Then complete your profile. When you complete your profile, please add your profile image, add social media links, add some background information about you so that we can all learn about you, and we can then help you identify topics that might be more relevant or interesting to you, and also assist you in other ways. Review the schedule of the program. So you saw how we have this curriculum tab, or we have a short summary on the main page. You can also reach out to us, and we're happy to share with you the syllabus for the whole program that shares a little more in detail what will be covered in each session. After you review the events, register for those events. So click register for the event, or you can also register for all of the upcoming events. And keep track of your progress. So we'll talk a little bit more in detail what that means, because we use a point system that helps all of us understand where we are and helps us align the progress of the whole group with what's actually happening, so that we don't run too fast and we don't move too slow. Here are a couple of good examples of people that have already completed a profile, and you can see how easy it is to understand who is who and start interacting with those people. So you can see that the person has a profile image. We assign different levels based on your progress. So essentially, the more courses you complete, the higher your level. We have a point system that essentially tracks all of your activity on the platform, including pipelines that you run, including courses that you complete, replies to forum posts, etc. And you can also see this badge that tells us that this person is a registered person of a program. So a little background on the OmicsLogic programs. We run 7 different OmicsLogic programs throughout the year. This is one of them, focused on data science, but we also have programs dedicated to genomics, transcriptomics, metagenomics, epigenomics, and many others, including organizations and universities that run the program internally. So here, for example, you can see the Louisiana Biomedical Research Network. So this person is a member of that program. Now you can also see what courses the person has been working on and how far along they are. You can see the completed ones, and it's important to complete a course so that that is registered. But also you can see the person's projects that they're working on and activity in general. Okay, if you have any questions about how all of this works, we welcome your questions. Please post them on chat. We'd be happy to answer them throughout this session or later on. You can also see a chat box on the website, so you can contact us through there. So what we are trying to set up here is a process that helps you get from where you are and advance so that you will have a sense of accomplishment. You know what you have achieved. You know what kinds of topics you have covered, and also prepare you for real projects and data sets that you can analyze on your own. As a part of this environment, we have set up a leaderboard, and you can see some of the top performers on the site. You will become a part of the leaderboard once you cross the threshold of 3,500 points. You have to be registered for the OmicsLogic Biomedical Data Program to be a part of the data science specific leaderboard. And we'll talk a little bit more about some opportunities that come with that. We'll be introducing you to our network. If you're looking to develop a network of contacts around the world that can help you gain access to job opportunities, research opportunities, and understand other conferences where you can present your research and others. So that is one aspect of it. The other aspect of it is really being a part of competitions that we will be launching in the next several weeks. One of them is going to be related to this program specifically, to the reproducibility of research that you do using computational methods. And I'll talk about that in a little bit. Okay, so now we've covered some of the aspects of the program, including the schedule, how to get registered, how to get started with a variety of resources that we have on the platform. Now let's talk about the hands-on aspect of the program itself. So the hands-on aspect of the program is tied to those different tools and resources that we started talking about in the beginning. So let's start talking about each one of those one by one. First of all, the T-Bioinfo platform. Now, some of you already have access to the platform. You've requested access, or you are a member of the site or one of our programs. For those of you that have not, after this session, we will be sending you additional emails with your credentials. So if you still do not have access to the T-Bioinfo platform, you have to request that access from us or wait for an email that will be coming out for those that have registered for the OmicsLogic Data Science program. Now, this program is covered on this link, tbio.info. You can learn about the platform, the vision for the platform, the research and educational tools that can help you get started is on the edu.t-bio.info. And then the analytical platform itself is on server.t-bioinfo. So let's go there. And thank you, Pratim, for posting that in chat. So if you look in chat, you will see the link to the platform. So let's just go over what is going on the platform, what it is, and why it is being included in this program. So once you gain access, you should be able to see your email address is your login, and you should receive an email with the password. Do not necessarily use the password that you have for the educational site. It's a different account. And you can see that once you're logged in, you will see a number of different sections available on the platform. So briefly, there is a section on next generation sequencing data, there's a section on mass spec data, structural biology data, and then data integration and modeling. In the context of this data, we will talk about all of these different sections, including processing of data, analysis of this data, and visualization of this data. And we'll talk about the connection between the various properties of data that we are analyzing with their biomedical interpretation. If you already have your login information, if you're already in there, you will be able to follow along with the demonstration that I'm doing now by using the demo pipelines. Throughout the program, we will not be using the demo pipelines. We'll be actually uploading our own data sets, and we will give you access to larger data sets that is going to be challenging to upload and download if you're just working from home. So we're going to try and make sure that anyone that's participating in this program can practically analyze these data sets, and we'll explain the process of gaining access to the platform as well as those data sets as we move along throughout the program. So we talked about the platform and what it is, and I want to talk a little bit about the challenge of these big data sets that are unstructured. So as you know, next generation sequencing is a process of shotgun sequencing. In other words, a variety of different portions of DNA or RNA are transferred into a sequence of nucleotides. And then our challenge is combining them together and aligning them to a reference genome so that we can, for example, quantify gene expression. So to quantify gene expression, you need to align those reads to the reference genome and then apply a variety of different tools to identify gene locations, locations of potential alternative splicing events that generate different isoforms, and then quantify those so that we get a table of expression. For those that are joining now, how do I request access to the analytical server? So those of you that have already completed your registration for the OmicsLogic program, you will be getting an email later today with your credentials. For those of you that have not completed, please complete the registration to the program by going on the registration link, and then we will be sending you the credentials. If you just want to explore the platform and understand how it works, you can also request separately by email a demo version of the platform. So let's take a look at the demo version and understand how it works. Once you're logged in, and you see your login information right here, you will see that by clicking the drop-down arrows, you get access to some demo pipelines. So we can go into the cell line pipeline, and what is important to understand here is that this is an interface, a visual interface, that allows us to connect a number of different scripts and algorithms with each other into a single pipeline. So this is what I want to demonstrate, but I want to also, for everybody to understand that each button here represents an algorithm, and you can learn more about the algorithm as the pop-ups come up, and you will see that in a second. So let's get started. This is a section dedicated to next-generation sequencing, analysis of gene expression, or transcriptomics. So transcriptomics involves several different steps: pre-processing, mapping, and then quantification of expression levels. This could be achieved using several different pipelines, and we'll start with the most simple one. We will go from start to a couple of pre-processing steps, then mapping on transcripts, and then quantification. You can click on OK. And now you can see that the platform will suggest some next steps. This is a demo. So in the real world, this will be based on the data that you upload and some of the settings that we have on the platform. So I'm not going to go into detail about each one of these steps because we have a whole session dedicated to processing of next-generation sequencing data, but I want you to notice that you have an explanation of each one of these algorithms, and for most of them, there's also a link to the publication. Now, as you go through, you will notice that there's also input and output files that you can take a look at to understand how each section works. As we map on transcripts and prepare the level of expression, the output will be a table where we see genes and samples, and for each sample we have a quantified level of expression. You can read more about transcriptomics in this series that includes transcriptomics 1, 2, 3, and 4. And you can see the link right here. So you can click on this link. And this link points you to some free resources that anyone, whether you're a part of this program or not a part of this program, can start working on. So transcriptomics 1 will talk about processing the data. Transcriptomics 2 will talk about differential gene expression. Transcriptomics 3 will introduce some of the dimensionality reduction, clustering, and classification algorithms. And transcriptomics 4 talks about single-cell transcriptomics. So you can see that the platform and the educational resources are closely integrated. And this close integration allows us to both help advance the whole program so that everybody covers the same material, but also keep track on where the challenges are and where do we need to spend more time. So the platform, again, look at some of these links. The platform will help you process large data sets, not on your computer. This is all done in the cloud, so you access the platform through a web interface, meaning that you can do the analysis anywhere, and it's not going to stop you from doing other things. But also, we have prepared some sample data sets that you can start with, play around with the demo pipelines, but also look at some exemplar projects that we have prepared for you. So you can create there a variety of different pipelines. Some pipelines very simple, and some pipelines are going to be more expanded like this. The goal of the platform is to help process large data sets, but also to do it in a very reproducible way. And I'll talk about this aspect of reproducibility towards the end, which is a very critical issue arising in a variety of different domains, but especially critical for those that are interested in more industrial level research. Excuse me. The second part of this program is going to be focused on R and RStudio. So we will use some of the scripts that are both on the platform, and then we will look at them in a more expanded way in your own RStudio environment on your computer. So to do that, and this is important for all of the participants especially, you have to download RStudio, install them on your computer, and test that everything is working. So here are some links. Let me go back. So here are some links where you can start this process. And please be ready for the next session with a downloaded, downloading RStudio, and R, and making sure that everything is working. Okay, so I pasted those links depending on your operating system, Windows, Mac, Linux. You can install the right R, and then once your R is installed and configured, go ahead and install RStudio, the free version, of course, and you will see there that you have a whole environment that helps streamline the process of data analysis and visualization. And we'll talk about how we can leverage a lot of different resources that are prepackaged, called libraries, that will allow us to do complex things in a very easy way. And now I just want to mention how these two are connected, right? So we talked about the T-Bioinfo platform, we talked about R and RStudio, and you're probably familiar with these, but I'm going to just show you how these two are connected. So if we go back to the platform, we looked here at how to process large-scale transcriptomic data. Now, once we have that table of expression, this is where a lot of times you would turn to other tools to start visualizing data, performing statistical analysis of data, training your machine learning models, etc. So under data mining right here, we have a couple of sections that we'll be using a lot throughout this program. So for example, we can go to unsupervised analysis, and I want to just demonstrate how we will connect the data that we have from analysis of, let's say, raw FASTQ data, all the way to data visualization in R. Whenever you are actually using the platform, not in demo mode, but in actual educational mode, you will be able to upload files. And here, we will be using next generation sequencing data. Let me quickly find that data set. Okay, so every time for a program, and yes, Roosevelt, the beginner can also take this course. All of this is going to be fairly simple. So what we will do in every session is we will provide you with a data set that we will be looking at throughout the program. So as you can see, it's a very user-friendly interface. All I did was I upload a data set, a TXT table. Now, after I press on continue, you can see that every step of the way I have some guidance, so I can understand what I am trying to do. And I have here 3 different options. I can do visualization using principal component analysis, clustering using a variety of clustering methods, and do network analysis. So in this case, let's try with something simple. We will use principal component analysis to simplify the data set that is complex because of the number of features that it has, in using simple graphical inputs to generate the analysis, but also download the R script that we can later on modify. Once I run my pipeline, you will see that the pipeline has started, and I have 1% because it has started right here. And I can see what are the steps that I took in my pipeline. So obviously, the more complex the pipeline, the more difficult it's going to be to remember every step of the way and what parameters were selected. So let's take a look at how this helps us create reproducible pipelines. Once I go to my pipelines, I will see a list of all the different pipelines that I ran, and I can see that my pipeline is processing. So you can see here that it's static, and to see the progress, I actually just need to refresh the page. I will also get an email notification to my email when the pipeline has completed. Beyond that, you can see here that simply hovering over the See More, I can see all of the steps that were used and what kind of data was used in the pipeline. After the pipeline has completed, I can also see a full report about my pipeline. So for example, if I look at, let me see, if I look at this pipeline right here, if it's not compressed, I will be able to see the report. So let me just, by the way, for educational pipelines, you will notice that every few weeks we compress the pipelines to save space. Okay, so let's take a look here. So you can see this is what a report looks like. It tells you when the pipeline was run and when this report essentially was generated, what is the name of the pipeline, what is the time it took to complete every step in CPU time and in real time, what are some of the files that were generated, etc. And you can see here a breakdown of this information right here. Now, this is very important for your analysis to be reproducible, because once you start working on a real project, you will see how each project takes some time, several weeks often, and so you will have to go back and understand, what did I do? Did I normalize my data? Did I just process raw data? What combinations of factors did I include here? And what were the settings for each step of the way? So now you can see that my latest pipeline is complete. It says done 100%. So let's go in here and let's take a look at some of the output files that this pipeline produced. So you can see here that the pipeline produced a number of different outputs, including a text file, a zip file of all of the results, and a plot, a PDF plot. So let's quickly take a look at this plot to understand what it means. Okay, so we have a PDF here. Okay, and the PDF looks like this. So the PDF shows us dots for samples, and you can see here some separation between obvious groups. So we will see that this PDF is actually produced by an R script. So let us download this R script really quickly to take a look at what's inside. This R script has the input data. It has the call that you would use in your terminal to call this script. And then it also actually has the script. So then we can open the script in RStudio and take a look at what essentially the script does, so that we can understand not only that we have an input and an output, but we also understand how that input and output has been done. So this is RStudio. And in RStudio, you can see that some libraries were loaded. You can see that some arguments were defined. You can see that we have loaded the data. So you can see that we are reading the table and loading the data. And we're also looking differently at the headers, at the rows. And we check for different names. And then you can see how we have a number of scripts that actually prepare the data. And we run principal component analysis. And principal component analysis is simply a dimensionality reduction technique that helps us look at the data from the best perspective that explains the most variability in our data set. Now, before I go too deep into this, I'm not going to confuse you right now, because I think we need several steps of the way before we actually jump into RStudio, but this is just to demonstrate that you can go from a pipeline on the T-Bioinfo platform, and then expand your capabilities in terms of visualization and more specific types of analyses. So essentially, the platform becomes as a springboard for you to start developing your own analysis and understanding the steps that you are making. Okay, so we talked about how to get started. And here's just an example that we will actually look at each step of the way and try to improve this principal component analysis. We will look at different R scripts that do clustering. We will look at R scripts that run things like random forest, or create specific visualizations like heat maps, box plots, etc., all in the context of combining large-scale data processing and preparation, quick data mining, and then looking at specific questions, and we'll get to that in a second here. How do I ask a meaningful question from this type of a data set? So we'll look at how we can actually go beyond the platform and create something a little bit more custom. In preparation for this type of analysis, I encourage all of you, whether you are participating in the program or you're just learning about the program, to take this free course. It's called Biodata Science. So if you go to the educational site, and if you're a part of the program, you will see it right here. If you're not a part of the program, just go to courses and just look at all the courses, and you'll find these courses. And here, let's just take a look at what this does. This is a very good tutorial that was prepared to introduce everybody to some of these topics. How to set up R Studio, how to load the libraries, how to check that you've got the right libraries, how to set up your environment so you know which directory you're working in. So all of the basics. Up to the point of loading the data, running principal component analysis, and modifying some of the look and feel of your results. So again, if you're interested in this program, you're wondering, how is this going to be a practical hands-on program for me? This is a good example project that you can go through, or a course, sorry, that you can go through, understand how it will work. So in the sessions, we will do this kind of guided demonstration. And then in your free time, whenever you have the time, you can go in and complete some of these assignments. Another good course to look at is this introductory course called Machine Learning for Biomedical Data. If you're not familiar with machine learning and you don't know the difference between supervised and unsupervised machine learning, what is clustering, what is classification, all of that is briefly covered in this introductory course. Okay, somebody asking here for a link to the Biodata Science free course. So let me just paste it here in chat. So this is the link. Okay. So once you get a sense for all of these different tools and what we are doing and how we are doing, one of the questions is, well, what are we doing? What is a meaningful project? What is a meaningful question for me to be able to address? And that is, I would say, a critical component of this program. I think there are plenty of courses out there that teach you about R and the different machine learning methods, but what is often lacking is, how do you ask the right question? How do you understand the problem in the context of the data that you have, and how do you choose the analysis methods to be able to answer that question? So let's take a look at an example. When we talk about biomedical data, we're talking about somebody that is a biologist or a clinician looking at the data and changing the way they interact with a situation, for example, a patient. So it could be somehow linked to this iceberg idea. You have on top clinical data, which is essentially clinical symptoms of a disease. Somebody comes in, something hurts, there's a growth, something on the surface. The challenge is to link what's on the surface with what is below the surface. And it's important to understand that oftentimes that is going to be a lot more deep than what's on the surface. So on the surface, you might have 20 different features, for example, cell count, or different elements that you can detect in blood or even visually. But underneath, you have this molecular detail, what's happening at the cellular or subcellular level, what genes are expressed, what modifications in the DNA, in a tumor are there. And then the question is, how do you link those things to the phenotype that we can interpret? So we'll talk about how this is done, but specifically we'll talk about it using practical examples. So this is one of the papers, and if you've been around through some of our programs, you will see that we use this example very often. It won't be limited to this example, but I think it's a good example that is impactful in terms of what it demonstrates, but also not as complicated as some of the other ones in terms of the amount of biology that you have to have in your background, and in terms of technical background that you have to have. So let's take a look at this. This is essentially an abstract from a paper called Modeling Precision Treatment of Breast Cancer, right? So this is just an example. And you can see down here that we have this link. Let me paste that in chat. Sorry. Okay, so we have this paper. And this paper, let's just quickly understand the highlighted words help us understand what it's about. So we're going to be looking at molecular profiles to identify features that can predict therapeutic response and select optimal predictors. Now, this is going to be a demonstration of molecular pathobiology that integrates omics level information and helps select most predictive molecular feature sets for a clinical classification in terms of diagnosis and selection of treatment. So you can see that in the results, the orange color indicates some of the technical methods, and the yellow color indicates some of the conceptual level for us to understand what they were doing. So they used least squares, support vector machines, random forest, copy number, aberrations, mutations, gene and isoform expression. So some of the data sets are described here, some of the methods are described here, but I think what makes a lot of sense is what we can see right here visually in this visual abstract. So you can see that they took a number of breast cancer cell lines, response to different drugs, molecular profile of those cell lines, and integrated that information to be able to predict response to drugs, but also they compared it to the Cancer Genome Atlas. So we can see that as an example of one of the challenges that could be addressed in the context of biomedical research and industry. So let's take a look at breast cancer. So here we have the mammogram, and the mammogram has this dot right here, right? So this is phenotypic information. This is something external, and we intuitively say, okay, well, it's a tumor. Now, it's a tumor that we need to categorize. We need to give more definition to this problem. So how can we give it more definition? We can, for example, look at the source tissue. Where did this tumor originate from? And we have these different names. For example, invasive lobular carcinoma is a name that describes this tumor from the standpoint of which organ or what types of tissue it originated from. Looking further at the tissue, we can actually take scans of that tissue, and we can stain it using immunohistochemistry to identify expression of different proteins. But when we talk about immunohistochemistry, we're actually having a limitation with the number of proteins that we can look for. So here you can see classification of subtypes of breast cancer using a variety of different proteins that are expressed or not expressed in a specific tissue. This is an example of molecular look at the same data. If we use multiple expression of genes or a gene panel, we can categorize the samples looking at their gene expression profiles. So here you can see how we can classify different subtypes of breast cancer using PAM50, 50 genes that help us understand in a more precise way how we can categorize these different subtypes. So these types of projects is what we will go into. And as you can see, here we have a variety of data sets. We have images, we have protein expression, we have gene expression, and we will try to link that to clinical questions. What is the survival? How will they respond to drugs? How do we assign them to a specific group of patients? So let's talk a little bit about what types of questions are there that we can answer using data analysis. Patient stratification. So patient stratification has to do with subtypes, has to do with identification of groups, well-defined groups, but also linking that to survival and recurrence. And patient stratification, you can probably see the link here to risk stratification. What is risk stratification? Is organizing our data in groups that can help us estimate the level of risk. The higher the level of risk, the more aggressive we can assign the treatment for that. And also, we can combine different elements of treatments that we have available to address what we consider to be the drivers of that risk. We can also look at responders versus non-responders, right? So when we talk about selection of treatment, we can look at whether a person will be responsive to a treatment, or is there some, for example, mutation in a protein where the molecule binds that will help us understand that that person is not going to be responsive to a treatment. This is especially important in the context of clinical trials. When we have clinical trials, one of the biggest challenges is selection of patients. How can we select patients that will respond or not respond to very specific treatments that we are trying to test, or combinations of treatments, right? Now, we also need to understand how we can use our molecular features to build predictive signatures. So this is called biomarkers or gene signatures. For example, we can look at gene signatures that can help us in predicting the outcome of treatment, that can help us understand what will happen far away in the future, for example, 5 or 10 years from now. We will look at target discovery. How do we understand when we analyze this molecular level data, how can we understand how different factors interact with each other to identify where are those critical points where we can intervene? And that's called the target. Which gene can we target? Which protein can we target? Can we silence a gene? Can we block a protein interaction? What can be done? We'll also talk about early detection. If there's a consistent process that drives the development of disease, can we early on detect that that process is starting? And can we also see the precursors of certain processes that we know eventually lead to an outcome that we want to prevent, or even know that it will happen? This includes risk. So we will look at germline mutations that can be associated with risk. And we'll look at also molecular profiling that can help identify the level of risk. Ultimately, predictive analytics, it has to do with machine learning, because when we look at training a data set, once we have labeled our data and we've prepared our data, we can use this data to predict something that will happen 2-3 years before it ever happened. And the more reliably we can predict that, the more valuable the machine learning method, the more valuable the analysis that we ultimately come up with. So we'll talk about predictive analytics in the context of this type of clinical and molecular data. Next time, when we meet, we'll start talking about next-generation sequencing and omics technologies. So we'll talk about how we can take raw, fast-queued data sets. How can we take genomic data, like whole genome sequencing, whole exome sequencing, whole transcriptome sequencing, or metagenomic sequencing? How can we take those different types of raw data and process them, structure them, so that they are ready for analysis? So that's going to happen September 18th. Again, at the same time, 9:30 to 10:30. All of you are invited to join. And hopefully that before this next session, you have already started making progress with that list. You have registered. You have completed your profile. You've gained access to the server platform. You've gained access. You've installed the tools that we will be using, including R and R Studio. You've opened up Excel. You've tried loading in some data in Excel and maybe tried to create some kind of visualization. So you're familiar and we're ready to go forward. I want to also briefly mention what this program will give you. So this program will cover methods for high-throughput data processing, analysis, and visualization, and can help you apply these methods to address clinical, research, and pharmaceutical challenges in the context of projects. It's a very project-driven program that will start from thinking about what is the challenge and how do we solve that using data analysis methods. It will help you design, prepare, and plan your analysis and make it impactful. So we'll talk about some of the challenges associated with data analysis and ultimately how to mitigate the risk that your analysis will not be effective or will not be clear, how to focus, how to clarify the problem statement, how to prepare and plan for something that might take several weeks in advance. We'll try and help you potentially build up a whole portfolio of projects and analysis examples so that when you're talking to your lab manager, to your PI, or planning your own project, you are thinking of it from experience and you can demonstrate your experience in specific projects. And will help understand biomedical research and read publications critically. I think this is very important. When we look at these illustrations, when we look at publications, at the analysis results that others have published, you saw how we were looking at those keywords, and we'll cover each one of those concepts so that next time you look at them, it all makes sense. You know each one of those words, what they mean, why they were used, and what alternatives are there to some of those methods. And finally, we'll for those of you that qualify, essentially participation here is key, that you join all of the sessions. The point system will help us understand who is performing well in the program, and you can get a certificate of participation that will be something that you can include in your CV, post on LinkedIn, etc. So this is a summary of the OmicsLogic program. A little bit of where we're going to be starting from. We talked about the schedule, we talked about the logistics, we talked about some examples of the hands-on activities, talked about some projects that we'll talk about. And finally, if you have any questions, now would be the time to ask any specific questions that you might have, or in general, if you would like to know about our team, about our programs. We have a lot of other programs that are ongoing, and so please go ahead. And now you can post your questions in chat, or also feel free to unmute yourself and please introduce yourself if you would like to ask any questions directly. So how long is this new course and how long will it go? All of our programs are around 3 months. So as I showed in the beginning, we will have 5 different sections of the program. So let's take a look. Where can you find this information? So if we go to the organization page, so I'll just paste the link here again, and I want to go over a little bit about the schedule again. If you go to the home page of this organization page, so you can see that right here you have upcoming events. So we started today, 16th of September, and then on the 18th of September we will talk about next generation sequencing, and this is just this week. And then as you continue to scroll, you will see these dots that show up on dates all the way to the 17th of October. By the 17th of October, we will be done with the basic part of the program. After that, we will focus on projects, which means that you will get one-on-one feedback on your project selection. You will get access to data sets that have been already prepared with some level of tutorials included, that you can start a project on your own and develop your own question around that data set and around that problem statement. Or you can just identify a meta-analysis project where you can look at publications and understand what others have done with some project so that you can think of your own challenge. Okay? So if you want to have a detailed curriculum, you can click right here on the tab, and there you will see all of the dates. So again, just scroll down and you will see all of the dates and all the individual events. And at the end, as I mentioned, we will transition to projects. So the full program will be around 3 months. So Pratim, thank you for and Darko, thank you for answering the questions about RStudio. This is an important question because, of course, you do have to have the updated version of R, and you also have to make sure that it works. So what I suggest is click on some of these links that we have in the chat and start installing R and RStudio, and to test that everything is working properly. If you go right here to courses, make sure that you check out this course called Biodata Science. So Biodata Science will help you test that everything is working properly. And it gives you some example that you can actually practically work through. Okay, so if there are any more questions, feel free to post them on chat, unmute yourself, and we will be happy to answer them.
Hello.
Yes?
This is Vamshi.
Hello, Vamshi?
Hello.
Yes, hello.
Yes, yes. I have a doubt on regarding. I have downloaded the Anaconda package version, so I can install RStudio from Anaconda package. So can I work through that?
Yes, of course. Yeah. So the way we will be going through the training is we will show you what libraries and what packages we will be using, and we'll also demonstrate it using specific data sets that we will send you in advance. So what we hope is that as you make the setup on your computer, you will be able to test it first to make sure that it works, because obviously there will be a lot of variation between how it's set up on your computer from somebody else that might be using a Mac or a Windows version. And so what we want to make sure is that we will address the conceptual questions and demonstrate practically that it works, and then you can look into how to make it work on your computer, and we can schedule even a one-on-one session to help you get through that. So the question here is, how much is the participation certification? So we'll be happy to email you that information. The list price is $200 for the whole program. Obviously, we have some discounts for students and others, so please reach out to us directly and we'll send you that information. Now, about the packages, there was a question. Thank you, Darko, for answering that as well. We'll walk through a lot of the packages. We'll obviously use some data analysis, some machine learning packages, visualization, and we'll talk a little bit more about which packages and why we are selecting throughout the program. Okay, so I suggest that we make this program again, the next session that we will have is going to be September 18th. Before September 18th, please follow through the process that we discussed here. Register, update your profile, make sure that you've got access to all of the tools. If you have any questions, we would be happy to answer you. We will start talking about next generation sequencing data processing in our next session on September 18th. And then we will go in how to visualize, summarize, and explore some of the data sets that we have prepared. Sylvia is asking link for packages. So we do not have a comprehensive list of packages that we will be using. We will start with a set of packages that you will find in the, again, so to get started, it would be very useful to get started with this principal component analysis example. And then in the example, you will see some of the packages that we start using there, and then we will expand from there. I mean, there will be a whole list. So if that's something that you're interested in identifying a whole list of packages that we will go through, we can probably put together a list and send it to you. So feel free to reach out by email or otherwise. So if you have any other questions, please let us know by email. You can reach out to us through the chat on the website as well. And if you have not gotten your access to the server platform, but you have already registered, wait for those emails. They will be coming later today. Thank you again for joining today. It was great to get to know some of the participants that are planning to join us. And if you have not completed the registration, you would like more information about that, please reach out. There are some email addresses right here in chat, Team Mohit, and we will be looking forward to seeing you next session, which will be on next generation sequencing data processing. Thank you.